Okay, so this is Lab 12 genetics, and we're going to start off with the notes because this is really just all theory. This lab, there aren't really too many images or anything to use, but we'll go and review what there is on the course page. All right, so there's about a dozen different terms to match up with functions, and so within the notes now you can take these and essentially do the matching A, B, C, D, E, F, G to the particular term. So chromosomes bears the genetic material and composed of DNA and proteins. We have genes that are the DNA sequence on a chromosome that codes for different proteins that determine an inherited trait. Allele is an alternative form of a gene. Dominant is the allele or gene that will be expressed and always identified with a capital letter. And recessive is the allele that's not expressed unless it's homozygous, which means that you have inherited two lowercase letters. Recessive is always indicated with a lowercase letter. Homozygous means the genes are identical. You can be homozygous dominant or homozygous recessive. So it's the term homozygous versus heterozygous below this. Homozygous means two identical letters. It doesn't tell you whether they're uppercase dominant or lowercase recessive. It just means, they're two identical letters. So homozygous dominant means two capital letters. Homozygous recessive means two lowercase letters. And heterozygous means essentially it's a combo pack. It's a combination of an uppercase letter and a lowercase letter. Right, then we have genotype and phenotype. Genotype are the actual alleles on a chromosome. So when we talk about the genotype of mom and the genotype of dad, is going to be the two letters, whether they're homozygous dominant, homozygous recessive, or heterozygous for a particular trait. And you can use whatever letter you want to represent. Let's say it just is a symbol. So using a letter B might be for blood type B. So you remember. And if you had two big B's, then that means that you're homozygous for blood type B. All right, so the genetic makeup of mom and dad is called the genotype. These genotypes are required to perform and fill out the Punnett squares. And then the combination of letters that come up in the Punnett square, are again your genotypes. This is the genotype of the offspring or the combinations of offspring, given mom's genetic makeup and dad's genetic makeup. And there's a good little video in there in the lab, some video links, about Punnett squares. And I would suggest you watch that because it's something that it's very difficult to do on a Zoom like this, but it explains what it is. And most people have done this before. They just kind of forgot how to, set it up. But when we look at the genotypes of certain outcomes of inheritance patterns, then given the combination of letters that came up in the, the Punnett square, the four squares there, you can either have 4 different combinations that come up. Therefore, your genotype would be 25%, of this combination came up, 25% of this, 25 this combination, 25 this combination, or it could be, hey, half of the, the two of the same genotypes came up in that Punnett square. So 50% are maybe heterozygous B, and 25% is homozygous B, and 25% is homozygous recessive or something. So genotype is also the percentage of outcomes, of the combinations that came up in a Punnett square. The phenotype is what you see, or the physical expression of what came out in the phenotype. So even though, let's say, when we look at blood typing, even though if you're blood type B, you could either have the genetic or genotype of either BO, which means you're heterozygous for B, or you could have inherited two capital B's, homozygous for B. Well, either way, you are blood type B. So the phenotype of those two combinations, would be, let's say, if all four came up in all the genotypes in the Punnett square came up with a capital B and something, capital BO or BB, then you would have 100% blood type B. But the genotype, the combinations were still maybe 50% BO and 50% BB, homozygous B. So yes, the letter combinations came up differently, so that has to be identified in the genotype, but the phenotype is the physical expression. Am I blood type B or am I AB, A, or O? Well, you'd be all blood type B. Okay, well, you'll get a better example of this as we pass through this. Sex length means it's passed through mom's genes. There are actually other sex length that are not necessarily passed through mom, but the majority of them, the ones that we're going to look at, are passed on mom's genes, which means it's passed on an X chromosome. Right. So the normal has two normal genes, a carrier mom, so we're talking about moms because moms are really, for the majority of these, this trait is going to be carried on an X chromosome, not on Y. So when it comes to mom's makeup, she can either have two X's that are just normal and not even be a carrier, or she could have one normal X and one X chromosome is carrying, let's say, CB for color blindness. Well, mom's normal chromosomes dominate over the chromosome carrying color blindness in females. So, even though her genotype is heterozygous for X with, carrying the chromosome, the color blindness chromosome, she will phenotypically, essentially, her physical presentation is going to be she's normal. Right? But she's capable of carrying it on to her offspring. So she may be just normal, she may be a carrier, she may have the trait. I will tell you that it's possible when we get there, we'll talk about this again briefly, is that women can have color blindness or hemophilia. If a woman inherited an X chromosome from mom that was carrying color blindness and dad was colorblind, and so she received his X, because you have to be XX to be female, then you could have a colorblind female. But that happens very rarely. Usually, the X is going to be passed on to the male. So for males, you need to be normal, which is a normal X or a normal Y, or a male could inherit an X carrying this particular trait and a normal Y. Well, the X is going to dominate over the Y and express that color blindness. Right. So that's what we mean by sex length. It's generally passed through mom's genes. All right, we can have homologous pairs of chromosomes. This is a pair of chromosomes that contain similar genes. So again, homologous, identical. Autosomes are chromosomes that determine general body characteristics, such as hair and eye color, versus the 23rd pair of chromosomes that determines gender. So autosomes are the first 22 chromosomes, determines general body characteristics, and the 23rd pair determines gender. Sex chromosomes determine the gender of the individual. You either have XX female or XY male. Okay, we have inheritance patterns. So not all of the things that are expressed in our body, things that we, different traits or characteristics or even disorders or abilities, like to taste certain things, are inherited the exact same way. We have these different ways in which we inherit. So we have what's called the autosomal dominant and autosomal recessive patterns. These are what's called the Mendelian genetics patterns. You either have something, you're either normal or you have a disorder. You either have the ability to PTC test, which is the ability to taste this bitter substance, or you don't. There's no in between. You're either one or the other. Those are your only two outcomes. So out of those Mendelian genetic patterns, we have what's called being an autosomal recessive pattern, which is the most common. And the normal gene, when present, is going to dominate over anything that's defective. So both parents are carriers. If both parents are carriers, or heterozygous for the trait. To have the disease, you must be homozygous recessive. So let me just back up again. Autosomal recessive pattern. You either are normal or you have a particular trait or disorder. And normal dominates over the disorder. And this pattern is the most common. In contrast, autosomal dominant below this is going to be the opposite. The disorder or the trait will dominate, and normal is going to be recessive. So in either case, whatever dominates is identified with a capital letter. So that's why in the autosomal recessive, if the normal gene is dominant, it means, let's say down below, we can talk about Tay-Sachs disease, right? So let's just use a T for that. Because normal dominates, then we're going to have, a capital T and a capital T maybe, or a capital T, lowercase t, because we said normal dominance. So maybe on the side, I might write, hey, big T, and then comma, big T, little t, equals being normal, right? Because the capital letter dominates, and we're now defining, hey, when then you see that capital letter, it means you're normal. To only get the disorder, Tay-Sachs disease, would be two lowercase letters, being homozygous recessive. So two little t's would equal Tay-Sachs disease. That's the only way you get Tay-Sachs disease. Now, in contrast, we can look at autosomal dominance. These are generally the more worse mamma jamma type of disorders that you can get. Frequently, you don't live long lifetimes, although some of these are managed a little more effectively now. The effective gene is dominant. Therefore, the mere presence of that gene, and it's going to be dominant. So we're going to use a capital letter to identify. In this case, our example is Huntington's chorea. We can use H's. You could either be homozygous for Huntington's chorea, big H, or if you've acquired a big H or a little h, the heterozygous form. Either of those two combinations come up, that equals you get Huntington's chorea. The only way to be normal is to have acquired two small h's or homozygous recessive. So then I might write to the side, little h equals normal. Once you identify what these different patterns mean, and once you choose, and we're talking about a particular disorder, Huntington's chorea, Tay-Sachs disease, PKU, then you choose your letters and you write out your three combinations, either two capital letters, two lowercase letters, or a combination of the two. And then next to it, put a little equal sign and go, okay, what is that going to mean in this inheritance pattern? If I get a capital letter, well, if normal, like in the case of autosomal recessive, dominates, then wherever the capital letter comes up, that means you're going to be normal. And the only way to get one of these disorders is if you have acquired like two of the lowercase letters, heterozygous recessive. And then the opposite happens with autosomal dominant. Whenever that capital letter comes up that dominates, shows up, and then that means you get the disorder. And the only way to be normal is to be homozygous recessive. You have acquired two of the lowercase letters from your parents. Okay, X-linked traits that are carried on sex chromosomes. Generally, the females are the carriers. For her to be a carrier, she's going to be heterozygous. She's going to have one normal chromosome, one normal X, and one X is going to carry a particular trait. The normal one always dominates over the expression of the X-linked allele. So, she could be, a female could have just two normal X's. She could have an X with an X carrying, let's say, color blindness, in which case the normal X dominates over color blindness. So she's still, genotypically, she might be heterozygous, but phenotypically, the physical expression is she has normal vision. She doesn't have color blindness. But she could have color blindness if she has inherited, an X from mom and an X from dad that carry color blindness. Males, have only one X, so it's going to be expressed if you have the involved X from the carrier. Right. So all they're saying is if you inherit a boy or a male, They get a Y from dad and an X from mom, and that X is carrying color blindness or hemophilia, then it's going to dominate over the Y, and it's going to be expressed. It may help to label the X's with, maybe an X with an N on the top to designate it's a normal chromosome, and then the other X maybe has a C for color blindness, H for hemophilia, B for baldness. You can make up whatever you want as long as you keep doing the same thing, the same pattern in the way you interpret the information over and over again. Okay, there's a codominant. And by the way, by the time we get here to the X-linked, the codominant, incomplete dominance, all right, that means that we are now looking at non-Mendelian genetics, which means there's a gray area in between. It's not just this, it's not just that. You can actually be kind of a mix of something, because it's got to be gray area. It's incomplete. All right, so the one that's best expressed, or I think is going to be the better example of this, is the codominant inheritance pattern. So our example of a codominant inheritance pattern is blood typing, how we determine the blood type, or what determines the blood type in you, which better said. Right? So both alleles are expressed if present. So I'm going to show you the exact part that is considered the codominant part. And this goes back to a review of, hey, what does it mean to be blood type A, B, AB, and O? All right, so this is it. If you are number one, blood type A, you are either have the genotype, the makeup of either having acquired or inherited two A's, two capital A's, so you're homozygous for blood type A, or you could be AO. That means you're heterozygous for blood type A. All right, so you're either homozygous for A, heterozygous for A. Either way, when you test those red blood cells, they have A antigens on the cell surface only. A antigens, no B. So you know it's blood type A. We're not talking about Rh positive or Rh negative. We're just talking about the ABO, AB international blood type A. Right. And I will tell you that A can dominate over O, and B will dominate over O. So to be blood type B, you are either or have inherited two B's, right? So you're homozygous for B, or you inherited a B and an O, in which case you are still blood type O. You have B antigens on the cell surface. B's dominate over O. So, those are two different forms genotypically, but phenotypically, the physical expression is you've got B red blood cells with B antigens on the cell surface. That's what determines that you're blood type B. Okay, or you could be blood type O, which means you've inherited two O's, and now you are blood type O. Remember, it means you have neither A nor B antigens on your cell surface. Right. Or you could be blood type AB. This is the codominant part. All right, so to be AB, remember when we were testing this in that first lab, we said, you're applying different antibodies. If I see thickening when I put B antibodies into an unknown blood, it means there must be B antigens on the cell surface, because B antibodies only bind to B antigens, right? So you saw what's called agglutination, that clotting kind of effect. And if I had more of that unknown sample, I put A antibodies in there, and I saw agglutination, that means there must be A antigens on the cell surface, because that's what produces the agglutination, right? A antibodies only bind to A antigens. So, that tells you I have both A and B antigens on the cell surface. That's what we mean by being codominant. That means you have inherited an A and a B, an A from one parent and a B from the other. Neither A nor B dominates over the other. Both of those antigens are present on the cell surface. So they share it. They both dominate over O. That's all. But they don't dominate over each other. Okay. Then we have incomplete dominance. This is a blend of kind of normal with affected traits. And our example of this is sickle cell anemia. Right. So I will tell you that normal dominates. right? So having the disorder, a full-blown sickle cell disorder, means you're going to be number 3, homozygous recessive, 2 lowercase letters that you've symbols that you've inherited. But I will tell you that the heterozygous form, that combination path, that person actually usually typically doesn't, they're asymptomatic. I mean, they generally don't have any symptoms of sickle cell anemia, but if you look at a smear of the blood, that you will see some cells that are sickling. Not a lot of them, but that they generally have at least a few, more or less, depending. And I will tell you that they can succumb to a sickle cell crisis, not maybe as severe as a full-blown sickle cell person, but if they're in the right environment to stimulate a sickle cell crisis, they'll start being symptomatic. So we have a totally normal person. We have a person that has a mild form, and if they're asymptomatic, they can then pass that on to their offspring, which we will call being a carrier. Or you have full-blown sickle cell, which means you are homozygous recessive for the disorder, and that would be expressed with 2 capital letters. So if we were looking at S's, and let's say we're going to use S's for sickle cell anemia, to be totally normal, so I might write capital S equals normal. Then I might write little s equals sickle cell anemia. And then the heterozygous form, capital S with a little s with lowercase s, equals mild form or just a carrier of sickle cell. And now okay, what does this mean? Well, how am I going to interpret the results of my Punnett square? All right, so Punnett squares are used to show the genetic possibilities of a couple, their genetic offspring combinations. We'll use a square with 4 boxes, with each box being worth 25% of your outcome, or 1 out of 4, 1 out of 4, 1 out of 4. You put mom's genes across the top, right, and dad's along the side, or vice versa. Some people put moms on the vertical y-axis and dad's on the horizontal. It doesn't really matter as long as you do the same thing every single time, because it's not going to change the outcome in the Punnett square. Then you insert the letters from each in the squares. So here they're saying it's not telling you what you're trying to determine, the offspring for, but they're saying, okay, so this person clearly on the top row would be heterozygous for B. We're doing blood, that's not even blood typing. It would be for, I don't know, whatever the B is going to be for. All right, blood type would be either big B, or big B, big O. So this is heterozygous parent for something, big B, little b, and also on the vertical, heterozygous for whatever B is trying to indicate the inheritance pattern or the genotype of. All right, so all they're saying, all this is meant to do, I wasn't very eloquent with that, but that the letters, you have a pair of letters that go on the top and a pair of letters that go on the vertical on the side. And then you take each letter and you draw it into the box. So here in the upper left-hand corner, I take a capital B and a capital B. On the lower left corner, I'd have a capital B and a lowercase b. On the upper right-hand corner, I have a capital B, lowercase b. And then the lower right-hand corner, I'd have 2 lowercase b's. The genotype would be, okay, how many came out homozygous dominant? How many came out heterozygous? Right? And how many came out homozygous recessive? Right? We had 25% came out homozygous dominant, 25% came out homozygous recessive. That's the lower right-hand corner. And then the upper right hand and the lower left hand, I'm sorry, are heterozygous, big B, little b, big B, little b. So 50% would be heterozygous, capital B, little b. Okay, so it says question number 1, mom and dad are both heterozygous for eye color of brown. Brown is dominant, blue is recessive. What are the genotypes of their possible offspring? What are the phenotypes? You can test yourself with that. So then these would be the outcomes. Here's another question 1. So these are the answers to it. So the genotypes are 25%, 50, and 25%, as I stated before. The phenotype is putting meaning to that. All right. So 75% would be brown eyes and 25% would be blue eyes. And the phenotype is the physical expression. I don't do eye colors in my labs or in my lecture classes, but the blood types for sure. Yeah. So remember, to be blood type A, you could be homozygous for A or heterozygous. AB, there's only one combination, AB. To be blood type O, you can only be O. And for B, you can either be homozygous B or heterozygous B. So now they're giving you a condition here. Mom is heterozygous for blood type A, and dad is homozygous O, or he's blood type O, because you don't even have to say it's homozygous, because to be O, you have to be OO and homozygous. But in any event, right? What are the genotypes of their possible offspring, and what are the phenotypes? So let's set up this Punnett square. We're going to put mom, says that mom is heterozygous for blood type A. So mom is on the top as AO, right, on the top horizontal. And dad was blood type O, so he's on the vertical on the left-hand side. Right. Now you bring those letters into each box. We have AO on the left, top left, and AO on the bottom left. And then on the upper right, you have OO, and on the lower right, you have OO. All right, so the genotype here is, well, look, we only had two combinations came up, AO and OO. So the percentage of AOs, half or 50%. And the percentage of OOs is also 50%, 2 out of 4. Mm-hmm. So we have the genotype is 50% AO, heterozygous A, and 50% for blood type O. The phenotype is, look, you are either blood type A, B, AB, or O. So 50% will be blood type A, 50% will be blood type O. All right, and that's the last of the examples here in the notes. Let me stop the share, and then let me go back, and we're going to go into the handout. Just takes a little bit. I mean, the lab guide. I always call them a handout. It's a lab guide. So we're going to go to Lab 12 Inheritance Lab Guide. And then I'm going to share that with you. There we go. Okay. So now you can go and match up the terms, right, with, the definition. Right? They're right out of the notes. Those are easy to do. Here we are in our Mendelian genetics, complete dominance versus autosomal dominant. Right? We have autosomal recessive inheritance pattern and the autosomal dominant inheritance pattern. Right? So we said, number one, the autosomal recessive means normal dominates over the disorder. Right? So you have to have that in mind. Right? And you either are normal, you have the disorder. All right, so it tells you mom and dad are heterozygous or carriers for PKU. So you can choose whatever letters you want, capital P and lowercase p. So you put those in for mom, and dad is also heterozygous, capital P and lowercase p. Put the letters in the box, figure out your genotype and your phenotype. Remember here, you were either normal or you have PKU. Being normal means you have two capital letters, you're homozygous or you're heterozygous. Wherever the capital letter shows up, it's going to dump. It means you're normal. The only way to have PKU is two lowercase p's. In contrast to autosomal dominant will be the reverse. Right? So I would write notes like that on the side. When you have autosomal dominance, the disorder or the trait dominates over normal. So the only way to be normal, is to have two lowercase letters. Right? And every other combination, whether homozygous dominant or heterozygous, because it's a capital letter, that means you get that disorder. So this is talking about the PTC testing. We're actually not able to do that right now because we are not in the lab, but tasting dominates over non-tasting. I don't taste it, so I must be homozygous recessive, for testing this bitter strip, this bitter taste on a piece of paper. But let's just move on to example 2, because this will make more sense. All right, so this is Huntington's chorea. So I would use maybe H's, and over on the side, I'd write H, 2 capital H's, right? Big H, little h, and then 2 lowercase h's. And then I write equal signs next to them, telling me what that it means if I have been inherited 2 uppercase letters, homozygous dominant. That means you have Huntington's chorea. If you have inherited a capital H and a lowercase h, equal sign, it means you have Huntington's chorea, or you're going to have Huntington's chorea. And then 2 lowercase h's equals normal. The exact opposite of what we looked at, right, in autosomal recessive. All right, because that helps you keep this all straight. Right, so mom has Huntington's chorea, Huntington's disease, meaning she's heterozygous. So, okay, mom on the top is going to be a capital H and a lowercase h. And dad is normal. All right, so he's going to be a little h and a little h. So you've already written it out to the side, so you know how to keep this straight. Right, so then you bring all the h's into those squares, figure out your genotype, right? And then the phenotype. What percentage has Huntington's chorea, and what percentage are normal? All right, then we move on to the non-Mendelian inheritance patterns. We have incomplete dominance. The example of that was sickle cell anemia. So we said mom and dad are heterozygous for sickle cell. All right, so again, over on this side, I might write capital S, capital S. That equals normal, because normal dominates. And these are the things you have to study. Capital S, lowercase s equals that you maybe have a mild form, or you are a carrier of sickle cell anemia. Or you could add lowercase, little s equals full-blown sickle cell anemia. All right, so now it says mom and dad are both heterozygous. So mom would be big S, little s. Dad would be big S, little s. Over on the vertical on the left-hand side. Bring the letters into the boxes. Do your genotype, do your phenotype. Now, your phenotype is either you are normal or you have Huntington's chorea. All right, then we have codominance. That's our blood typing. So it says mom is blood type O, so she's an OO on the top, right? And dad is AB, so you have A and B on the vertical on the left-hand side. Again, bring the letters into the boxes. Give me the percentage of your genotypes, and then the phenotype. Because in this case, look, you can either be O, A, B, or AB, right? Those are your combinations. Right, so then what would be the physical expression of the genotypes? Right, then we have sex-linked. These are the things that are carried on the X chromosomes. So you're going to have mom XX on the top and dad X and Y on the side. Right, they're going to tell you, and again, I would maybe write this over on the side. What is it? X2, 2X is carrying nothing. Nothing, not carrying any particular trait on it. Like this, like color blindness or hemophilia, would equal a normal female. And X, a normal X, and an X carrying, let's say, color blindness, would equal normal vision, female, but a carrier. And X with color blindness and X with color blindness equals a colorblind female, for sure. Those are the combinations, the 3, just for females. Then you could have an X and a Y equals a normal male. An X carrying color blindness and a Y equals a colorblind male. Now you know what the different combinations that can come up and what they're going to mean. All right, so it says mom has normal color vision, but her father was colorblind. All right. So that means, okay, dad has normal color vision and mom has normal color vision, but her father was colorblind. Okay, so let's start with dad and just put that on the vertical over on the left-hand side because you just wrote maybe over on the side, hey, for a male to have normal vision, he has a normal X and a Y. All right. So you just put X and Y on the vertical there over on the left-hand side. Well, what they're trying to tell you here with mom is that although she has normal vision, what makeup was she? Was she two X's and then one of those X's carrying color blindness? Or did she have two X's both carrying color blindness? Well, it had to have been one of the first two. Right? So she is the heterozygous form, right? Because she's a carrier and she's capable of passing it on, to her offspring. The only X she acquired from her dad was carrying color blindness. So even though, she got an X from her mom that was normal, the X she got from dad was carrying color blindness. But as we said before, a normal X chromosome dominates over an X chromosome carrying color blindness for females. So she has normal vision, but she's a carrier. All right. So on the top row, you put one X, with maybe CB on it for color blindness, and the other X with an N or nothing at all, because that's the normal one. And then you're going to fill in your squares. And then, essentially you can do your genotypes over the time. That's normal. What percentage of males, were colorblind? What percentage of colorblind females? What is the percent of normal males, the normal percent of females? All right, then we have exercise 4, karyotypes and non-disjunction. I generally don't do those either because by the time we get through all these different inheritance patterns, I usually don't have time for it. But I would like you to maybe look at those karyotypes because the karyotypes are a map of your chromosomes. And starting with like the largest ones to kind of the smallest ones, they're in pairs, pair 1, 2, 3, 4, 5, 6, all the way through 23. And I think it's helpful to look at those, especially to look at the 23rd pair, because that is the pair that determines gender. Those are your X's and Y's. And then whatever is on like the image, out of those images there. Okay, so let me stop the share. And I'm going to now go back and get into the images, because every chapter has some images. Let me go back into table contents. And then we will be done. This is your last lab before exam 3. Okay, so we'll be down to the bottom here. Lab 12. A few images. There we go. There we go. Okay, and yeah, I was, as I suspected, it's probably all this. All right, so these are some karyotypes. They're sideways, of course, but if you make it a little bit bigger, and usually we've done this in the classroom, but you may not have been able to see it. But that is sort of the mapping. You have to the upper left-hand side, you have chromosome pair 1, 2, 3, going across from left to right, top to bottom, ending with the chromosome pair at the end at A. And that is the 23rd pair that determines gender. They're rather interesting to look at. All right. Oh, that one's right side up. So this is a normal male, has a normal X and a normal Y. And it says normal male on the top of the chart. That's it. But that 23rd pair is the pair that determines gender. All right. And then we have here the 21st pair that's being pointed to here. Trisomy. There's something called aneuploidy. And when the chromosomes, remember that they're supposed to line up in pairs in the center during meiosis 1 and meiosis 2. You have these that line up in the center and then separate. So on meiosis 1, you have two pairs that line up next to each other, and one pair goes to one side and one to the other. But then in meiosis two, just like kind of what we really looked at in mitosis, is that you have the pairs, a single pair, or I want to say the 23 pairs line up right through the metaphyseal line there in a linear orientation, and they separate from each pair, in midline. Remember, meiosis one, we have what's called tetrads, where you have replicate all the chromosomes, but instead of them all lining up in the middle and splitting, and one goes to one end and one to the other, you have two pairs line up. So it's essentially four chromosomes in the lining up along the, in the during metaphase, and one pair goes to one end and one to the other. That's the portion that allows crossing over to occur. Right? So that ends meiosis one, and then we don't replicate, and we just line up all our 23 pairs, individual pairs, during metaphase, and now pull apart so that one member of a pair goes to one end of the cell and one to the other. And when you have, if they don't separate properly and too many chromosomes go to one cell and not the other, then you can have either too many of, let's say, in this case, we're looking at chromosome pair 21. You could either have maybe only one or three or even four. So, well, then, then we have more chromosome pairs. Yeah. Anyway, so in this case, what we refer to as trisomy 3, right? 3 chromosomes at the 21st, pair or location of chromosomes. Trisomy 21 is associated with Down syndrome. That can also happen, aneuploidy, where you inherit either, a Y and no X, an X and no Y, two X's and a Y, maybe two Y's and one X. You can, something went wrong when it came to the splitting up, of those chromosomes. If you've acquired like this, an A, right, two X's and a Y, right, that's called Klinefelter syndrome. That's what's on the top of that chart. Or here we have Turner syndrome. You see at the last pair, you have only a Y, an X and not a Y. Then that's called Turner syndrome. Okay, so that is the end of the lab and the notes. Thank you.